So in previous video we discussed about the sexual reproduction and we discussed about the conjugation, right? So now we will discuss about the transduction. Now basically what is transduction? Basically in this there is transfer of genetic material through the phages, right? Basically bacteriophages, right? Consider that over here we are having a bacteria, right? And this is basically a phage, right? And this is bacterial chromosomal DNA. As it will enter, it will insert its own DNA, right? So this is the bacterial DNA and this is the virus DNA. After this, what will happen? There will be the process of assembly, as we have discussed this in lytic cycle 2. Like there will be a lot of bacteriophages. But sometimes what happens, like we know that basically when virus enter, like virus genetic material enters into the bacterial cell, it hijacks the machinery of the bacteria. But sometimes what happens, the bacteria's DNA gets into the bacteriophage. Sometimes it happens. So sometimes, for example, consider this is the bacteriophage and it contains virus genetic material plus the bacteria's genetic material. Now what will happen when there will be the realization, when there will be the process of budding, like when the cell will blast? This is the bacteriophage, right? This contains the bacterial genome as well as the virus genome. When it will enter into, and like it contains both, when it will enter into another cell, there it will carry that bacteria's DNA plus its own DNA too. This is how there is a transfer of genetic material through phages. Like this bacteria's genetic material is now in this bacteria due to this phage. Sometimes it happens too. Now third is basically transformation. Third is transformation. Basically sometimes, like this, for consider this is a bacteria and there is a capsulated bacteria. Like this is a capsulated bacteria. But sometimes what happens in adverse conditions, it loses basically its coverings. It loses its covering, its cell wall, its cell membrane, all, and its capsule all get lost due to the adverse condition. Now what will happen? This genetic material is surviving. Now consider that there is another bacteria which is non-capsulated. This is non-capsulated bacteria. This is non-capsulated and its genetic material. Now what will happen? This bacteria will attract this genetic material towards itself. This is only a genetic material, as it has lost its covering, its capsule due to the adverse condition. Now what will happen? This genetic material will enter into this cell and there will be the process of recombination. These both DNA will recombine to each other and they will form one genetic material. Now what will happen, as this genetic material has combined with this genetic material, now a capsule will be formed on this bacteria. Why? Because it has transmitted this bacteria's genetic material into itself. So obviously what was the characteristics of this bacteria? That there was a capsule. So but this was a non-capsulated bacteria, so obviously it will develop that characteristics and it will also develop the capsule in itself. So here the DNA is injected. So I hope this is clear to you. If you have any kind of queries, you can ask me in the comment section. I will be very glad to answer you guys.